In this video, we're going to talk about how viruses actually infect their host and how they can make their host sick.
Viruses have cytopathic effects. And if we look at this word cytopathic and break it down, cyto refers to cell and pathic or pathos refers to disease. So viruses actually cause disease by infecting cells. And viruses are intracellular parasites. So once again, intra means within, and cellular means cellular, so within a cell. And they are parasites in that they are not able to live and reproduce on their own. They have to use their host cell's resources in order to make copies of themselves. And in the process, they harm the cell because they're using the cell's resources. They're using the cell's energy, raw materials such as things like amino acids or nucleotides, also using their organelles. So the mitochondria are now busy making ATP for the virus instead of the cell. The ribosomes are busy making proteins. And these are all things that the host cell is supposed to be making for itself or for the multicellular organism that it's part of. But now the virus is taking over all of these resources, and the cell is not able to perform the functions that it needs to perform. Some examples of cytopathic effects of viruses can include lysis. Lysis means to burst. So essentially, a virus can ultimately cause a cell to burst or explode. Apoptosis just refers to cell death. So this is another way that the virus might kill the cell, releasing viral particles in the process. And not all cells infected by viruses die, but remember that their function is being altered. So they are still being harmed in the process because the cell is no longer able to perform its normal functions. Now, interestingly, a lot of the symptoms associated with the viral infection actually have to do with the host organism's immune response. So, if we think about humans, viral infections can cause things like fevers, fatigue, sneezing, coughing, etc., etc. And these are all ways that the human immune system is trying to fight off this virus. So by inducing a fever, it's trying to make the environment less conducive for a virus to survive and reproduce. The fatigue comes from the energy that your body is using in order to make immune cells and antibodies to fight off this virus. Things like sneezing and coughing or even diarrhea are ways that your body is trying to get the virus out of your body in order to get rid of it. We can break down the steps of a viral infection into kind of four main steps. And we are going to talk about how these steps can differ depending on whether the virus is a bacteriophage, which remember bacteriophages are viruses that infect bacteria, but we also have viruses that infect animals or plants. And also remember that viruses come in different varieties. So some might be enveloped, meaning that they have that kind of outer membrane that surrounds the capsid, or they might be naked or non-enveloped. And all of these things determine how this virus is able to infect the cell.
The first step of a viral infection is the attachment stage. Now, we've talked about how viruses have some specificity to them, and that specificity has to do with the type of organism that it can infect, but also the type of cell that it can infect. So, for example, if it's a human respiratory virus, then that virus is only going to be able to infect humans, and it's only going to be able to infect cells associated with the respiratory system. This is one reason why plant viruses cannot be transferred or transmitted to humans and vice versa. And the specificity comes through this kind of lock and key or puzzle piece model. So if we look at a cell here, okay,
And your cells have these protein receptors attached to their surface. And these protein receptors might come in different shapes and sizes. And I'm making just some, real basic shapes here, but they're much more complicated than that. And these receptors are often made of proteins, but they also often have carbohydrates or maybe even lipids involved. Now, viruses, if you recall, we talked about how a virus has kind of a DNA or RNA core, and then this capsid around it. And then some of them are enveloped, others are not. So either the capsid proteins, or I'm going to go ahead and draw an envelope here. So if the either the capsid or the envelope is going to have these proteins, we talked about the spike proteins in a separate video associated with something like coronavirus. So these spike proteins have to match the receptor in order to attach to the host cell. So in this case, you would have the host cell with this specific receptor that's kind of round shaped. And then of course I drew this virus much larger than it is. In reality, it's very, very small. But this would be like one of the little spike proteins attached to the cell. All right, so the virus has to attach to the cell this way before it can actually get into the cell.
the second step of a viral infection is entry. So once the virus has docked with the host cell, so here these blue circles are the host cell. And remember that this kind of line here, this is our phospholipid bilayer. So this is a cell membrane just like what you probably learned about in a first semester biology course. So once the virus attaches to those receptors on the host cell, some or all of the virus is then going to go inside the host cell, so into the cytosol or cytoplasm.
In this top picture, we're looking at a bacteriophage. And so that would make this blue cell a bacterium. And the bacteriophage just injects its nucleic acid. So this squiggle right here is going to be the viral DNA or RNA. Now, if a virus is naked, meaning it does not have an envelope, we're going to get a situation like this here. So this orange thing is the virus. And the host cell's membrane is going to go through endocytosis. Remember that endocytosis is a form of kind of transport where the cell uses energy in order to basically wrap its cell membrane around something, in this case a virus, and it actually pulls the virus into the cell. So that virus is going to end up in a kind of in a vesicle inside the cell. And then once it's in there, the cell is going to do different things to then access the genetic material that's inside that virus. And then last but not least, for an enveloped virus, we have this situation over here where the envelope or the membrane of the enveloped virus, and that envelope is this area here. And remember that this envelope is similar in composition to the cell membrane. So this envelope actually just fuses with the cell membrane and it releases the virus itself into the cell. And then once it's inside the cell, then you have the cell is going to do things to break down those capsids and get access to that genetic material inside the virus. Once the virus, or at least part of the virus, has been injected or inserted into the cell, the virus is then going to use the host cell's resources to replicate itself. And with replication, we're talking about the virus needs to make proteins, more nucleic acid, so it has to replicate its genome. It might need to make some enzymes, it might need to make some other molecules, because essentially it has to make a copy of its genome, it has to make new capsids, it might have to make a new membrane, and these are all things that it is going to do when it's inside the host cell. And in the process, it's going to use the host cell's resources to do this. So it's going to use free nucleotides in order to make the genome. So remember, our nucleotides are things like adenine, guanine, cytosine, thymine, and if it's RNA, we have uracil instead of thymine. The virus is also going to use the host cell's enzymes. It needs those enzymes to make ATP, which is the energy part here. So this could be ATP or other energy molecules that it's going to use for the cell to make ATP. But these enzymes are also needed for that replication process. So to make more DNA or to make RNA, you need things like DNA polymerase or RNA polymerase. These enzymes are also needed to make proteins, amongst other things. And then also amino acids, because remember that these are the monomers of proteins. So remember, the host cell needs these amino acids to make proteins for its own purposes. And so this virus is coming in and basically taking these raw materials and using it to replicate itself instead. The virus is also using the host cell's resources in order to make these different molecules that it needs. So for example, the mitochondria, it's going to use it to make ATP. The ribosomes, it's going to use it to make proteins and enzymes. And then the endoplasmic reticulum and the Golgi apparatus are also involved in making and packaging those proteins. So I like to think of a virus as kind of like the worst friend you've ever had. So imagine if you have a friend that calls you up and they say, hey, I really need to make some cupcakes for a bake sale at school. Can I come over and make them at your house? And you say, sure, come on over and make some cupcakes. So they show up and they have no supplies of their own. So they use your flour, your sugar, your eggs, your cupcake pans, your cupcake liners, your oven, your electricity, etc., etc. And it uses all of your resources to make these cupcakes. And in the meantime, now your kitchen is a mess. Your oven is busy, so you can't make dinner for yourself or your family. You're out of cupcake making materials. You're out of sugar. You're out of flour because your friend, quote unquote friend, has used all these things to make their own cupcakes. And so that's kind of how a virus works. It kind of goes into this cell and basically uses all of its resources for its own purposes. And then the cell doesn't have what it needs in order to do what it normally is supposed to do. Continuing with this third step of replication and assembly, let's talk a little bit about how the different types of viruses do this step a little bit different from each other. So remember that the genome, or the genetic material of the virus, can come in different varieties. So some viruses have DNA as their genome, some have RNA, and then we're going to talk about this thing called a retrovirus here for kind of the first time. Now, regardless of what the viral genome is, the ultimate goal is that the virus needs to make new proteins. Remember, those proteins are made for or needed for things like the capsid and maybe the spike proteins. Could also be enzymes, etc., etc. And it also needs to make copies of its genome. So it has to make more DNA or RNA. So it's essentially, it has to remake all the components of itself in order to make new baby viruses. Now, if you go back and think about this kind of this process of protein synthesis that you've probably learned about before. So protein synthesis is just, synthesis means to make. So protein synthesis just means to make proteins. And we can divide this up into kind of two overarching steps. We've got transcription. And then after that, we have translation. And you've maybe seen this actually drawn as DNA, arrow, RNA, more specifically mRNA, and then protein. And then this first arrow is the transcription arrow. And the second arrow is standing for translation. And remember that in order to make a protein, we need ribosomes. Okay, so going back to our different types of viral genomes here. So if the virus has DNA as its genome, the first thing that it has to do is transcribe that genome into our messenger RNA. So remember that M stands for messenger RNA. And then that messenger RNA goes to a ribosome in order to make those proteins. The DNA is also going to make copies of itself using the free nucleotides and enzymes and other structures in the host cell in order to make more copies of itself. Now, if the viral genome is made up of RNA, it might have to make a complementary strand of RNA before it can go through the transcription and translation process. And remember that complementary just means that if the original RNA sequence, let's say, is adenine, cytosine, guanine, uracil, the complementary strand, remember A always pairs with T when it comes to DNA, but in RNA, A always pairs with U, C always pairs with G always pairs with C, G with C, and then U with A. So this would be our complementary strand, which could then go into being transcribed to make mRNA, which once again is going to make proteins. Now, some RNA viruses don't have to make this complementary step, and they can just go straight into the transcription phase. It just kind of depends on what type of RNA it is and what type of virus it is. But once again, the goal is to make viral proteins. And then it also makes copies of the original genetic material in order to make new genomes. Now, retroviruses are especially interesting to me in that their original genome is RNA, and they actually have the ability to transcribe or basically convert that RNA into DNA. So we usually think of DNA as being a one-way street with DNA being used to make RNA. But in this case, these retroviruses can actually kind of go in the opposite direction. So they actually use a special enzyme called reverse transcriptase. And remember that enzymes often end in this ASE suffix. So that's kind of one clue that this is an enzyme. So these retroviruses use reverse transcriptase to convert their RNA genome into DNA. And then that DNA actually fuses with the host genome. So it's incorporated into it. So if the host genome is, let's say, a chromosome, then the reverse transcribed DNA of the virus is actually going to become part of the host genome. And then in doing so, that DNA that now includes the viral DNA is going to then cause the synthesis and assembly of new viruses. The fourth and final step of viral infection is going to be release, because the ultimate goal is for the virus to spread and go infect other cells or even other organisms. Now, there are a couple of different ways that these new viral particles are going to be released. So one way is known as budding, and budding is where the viral particles are released from the host cell, and it doesn't directly kill the host cell in the process. It is still harming the cell, so it doesn't kill it, but it does harm the cell in the process. And that's because, remember, cells, all the cells in your body or the cells of any organism, those cells have functions. They are doing things to keep you alive. And now the cell has been taken over by a virus, and it's been turned into a virus replicating machine. And so now the cell is not able to perform its regular duties, which is going to harm the organism. Remember that these viruses are using the resources in the host cell for its replication. So it's using up nucleotides and proteins and energy, and keeping the organelles busy so they can't do what they're supposed to do. Now, another way that a virus can release its newly formed viral particles is through lysis or apoptosis. Now, lysis means to burst. Apoptosis just means cell death, but kind of the same thing happens. Either way, the cell is going to die, and in doing so, the viral particles are going to be released. So if we go back to the example of your friend coming over and making cupcakes and using all your resources, budding would be like if she basically set up a cupcake store in your house and just constantly came over, used all your resources to make cupcakes that she then carried out of the house and delivered to different areas. Lysis or apoptosis would be like your friend coming over, making cupcakes using all your resources, and then basically exploding your house or burning it down, throwing cupcakes everywhere in the process. So these are different strategies. Lysis basically allows the virus to release a whole bunch of particles in a very short period of time. So it's kind of the shotgun strategy of, let's just release all these viral particles, and hopefully there is a host organism or host cell nearby that those viral particles can then infect. Budding is more of a kind of a long-term strategy where the virus is just constantly releasing these new viral particles, and in the hopes that, if there's not a host cell available right now, there might be one in the future when another viral particle is released. But it's usually not as widespread of a release because you have individual viral particles coming out of the host cell at a time. So let's take an example of a viral infection that affects humans. So in this case, we're looking at the flu virus or influenza. So you can see in our little picture here that we've got our virus here, and it's got the little spike proteins attached to the surface. And those spike proteins have bonded to receptors on the surface of the cell. So it's not really showing you that, but this line here is the cell membrane. And there would be little receptors sticking out of it in order to allow that virus to dock. So after that happens, the host cell, so remember this is the host here, and this is the virus here. So the host cell is going to engulf the virus by endocytosis. And so now this virus here is inside a vesicle inside the cell. Once it's inside the host cell, it is going to essentially release its genome. So it's going to release its RNA into the host cell. The viral RNA then enters the nucleus of the host cell, where it is replicated by RNA polymerase. So remember that this is an enzyme that is involved in making RNA. The viral mRNA is then used to make viral proteins. Remember, this happens through that transcription and translation. And so you can see here that the host cell is essentially making copies of the viral genome and of the proteins. And remember, these proteins might be associated with the capsid, or they might be those like those spike proteins sticking out of the envelope. They might be enzymes, etc., etc. And then the last stage is where all of these particles come together in order to make new flu virions. And this is an example of budding, in which the host cell is releasing these viral particles into the extracellular fluid. The cell is not killed in the process. It's just going to keep making new viruses until hopefully the immune system can shut the whole process down. Here we have an example of a bacteriophage infecting a bacterium. Remember that a bacteriophage is just a virus that infects bacteria. So you can see here starting out that the phage infects the cell. So it's going to inject its genome into the cytoplasm or the cytosol of the bacterium. Now, interestingly, the DNA from that bacteriophage is actually going to become incorporated into the bacterial chromosome. So remember that bacteria have a single circular chromosome that makes up their genome. And in this case, this genome now includes some of that viral DNA. Now, when bacteria reproduce, they go through binary fission. And remember that binary fission itself makes clones or exact copies of the original cell. And so every time that bacterium goes through binary fission, that viral DNA gets copied along with the genome of the bacterium. And this can happen again and again and again. So here it's showing one cell forming two cells. These two cells can then do binary fission to make 4 cells, which can make 8 cells, which can make 16, et cetera, until you have this kind of exponential growth of bacteria that all have this little piece of bacteriophage gene or genetic material incorporated in their genome. This process is known as the lysogenic cycle, and I apologize, on the slide I have it mislabeled. So this should actually be the lysogenic cycle at the top here, and then we'll talk about the lytic cycle down here. Now, under certain conditions, the prophage, or the DNA from the bacteriophage, can actually be removed from the bacterial genome, and the DNA replicates and these phage proteins are made. So essentially here, now we are making new viral particles. So new virions are being made. And those viral particles are assembled inside the cell. Remember, it's using the cell's raw materials and energy and organelles to do this. And then the cell is going to lyse, which, remember, lyse means to burst open or explode. And in doing so, all these new bacteriophages are going to get released into the environment to go find other bacteria to infect.